Thank you for coming to us.
Oh, yeah. Thank you very much. Can everybody hear me? Yeah? Okay. Yeah, I got a little sore throat here, so I'm going to be a little soft. Just let me know if I'm not speaking loud enough. You can seriously hear this? Really? Okay. Doesn't sound like I'm talking very loud. Thank you very much for that nice introduction, and thanks to CCMB for inviting me and for funding Clay and I. The money is supporting this kind of research, and so this is like a progress report on how the project is going. So I'll talk about functional connectomics and biomarkers in psychiatry, and I don't know what your format is, but I think I have like an hour or something like that, and rather than do like questions at the end, if you'll ask questions while this is going on, I think that'll be better. So that kind of format, just raise your hand whenever. So biomarkers, what are they? The theme here is that psychiatry, there's a worry that it's kind of a squishy field, and you know, we do interview individuals, and we don't have objective criterion. And so biomarkers are the idea that we need some sort of quantitative indicator of the disease process or pathogenic states, pharmacological response. And there's much interest in psychiatry for identifying biomarkers. It's essentially like the buzzword to get grant funding in psychiatry. You stick biomarkers in the title of the grant, in the specific aims, in the methods, and the resources, and in the conclusions, and you'll get funded. So here's a kind of flow process of, in schematic form, of how individuals come into psychiatric care. So this individual feels sad, goes to their psychiatrist. I don't know if people are familiar with the DSM-4 manuals. They're the Bible for identifying psychiatric diagnoses. And the psychiatrist, based on an interview, makes a decision. Well, does this person have depression, or do they have bipolar depression? It's very hard to tell from an interview whether it's bipolar depression, which is a specific type of depression with very different prognostic significance and treatment implications. So if we had quantitative markers to replace things like a clinical interview, then we would be able to sort people in diagnostic categories. And the implications, you know, if I decide that the person has depression, do I send them to the therapist? Do I give them medicines? Which medicine do I give them? We also want biomarkers for treatment response. We want to know, should they get this treatment or that treatment? If they get that treatment, what is the prognosis? Hopefully a biomarker could help to identify that. Beyond that, there's biomarkers of disease vulnerability. So this person, back when they were smaller, younger, and slightly different shade of yellow, they don't have the sad face that they have here, but they may have, as a latent quality, some sort of depressive quality, and that might be objectively discernible via biomarkers. Imaging has long been thought that it's thought that we could furnish biomarker for psychiatry. And the classic way that this is done is you take healthy individuals, people with different disease categories, like bipolar and major depression, you put them in the scanner, and you have them do a task. And this task is look at neutral faces versus harsh faces. Excuse me. And then here's the famous amygdala, and you can see that in bipolar depression, it was lighting up, whereas in major depression, it was not, and in control individuals, it was not. At one time, I think it was thought that these kinds of tasks and looking at individual discrete brain regions, we would find the biomarkers we need in psychiatry. But the fact is that even when we find statistically significant differences in an individual brain region, the amount of discriminative information we're getting is not that substantial, and the effectiveness of these kinds of things for classification is not that good. So we need to find more sources of information from functional imaging. And what I'll be talking about today is a method to get more information from fMRI scans, and this is Connectomics. So what is Connectomics? Well, everybody's putting omic after different kinds of things. So one justification for Connectomics is we want to join that kind of bandwagon. But there's actually a good scientific rationale here. Some people here may be familiar with C. elegans, the worm, and it only has like 500 neurons, and every single axonal connection between neurons has been mapped and has been known for a long time. That's structural connectivity. That's the pattern of wiring in the brain. That's not what we're talking about today. We're talking about functional connectivity today. Functional connectivity refers to the way the brain regions, quote unquote, talk to each other. It's a kind of a broad term we use for functional activity that's coherent between different brain regions as measured by functional scanning as opposed to structural scanning. The other thing that I'll often talk about is intrinsic functional connectivity. For now, what that means is the kinds of connectivity signals we're observing are not driven by task, not by presenting harsh faces versus neutral faces. There's something that the brain spontaneously does irrespective of task. So let me explain this idea of intrinsic functional connectivity more. People are very familiar with this idea. In the scanner, we give people pictures of harsh faces, and then alternatively, we give them pictures of just a cross on the screen to observe. And we measure the bold signal. The bold signal is the signal that's being emitted. Another way to put it is it's the signal that's detectable by standard fMRI scanning. And what the bold signal is doing is during the neutral condition or the cross condition, it's staying quiet in this particular voxel of the brain. It's elevated when you show faces, and it goes quiet again when you're just showing a cross. That's the activation perspective where you're driving the bold signal based on the stimuli you're presenting. The connectivity perspective is different. The connectivity perspective, for at least the first few analyses I'll be talking about, we're not showing people any stimuli at all. They're just in the scanner and they're looking at a cross. And you might think, well, if they're just not doing anything, the bold signal is just going to be quiet like that. Well, that of course doesn't happen because you'd be dead if the bold signal is just not doing anything like that. Here's what's really happening. The bold signal is oscillating. It's exhibiting spontaneous fluctuations at about 0.01 to 0.1 hertz. That phenomenon has long been known, that the bold signal is oscillating like that. That's been known for about 15 or 20 years. What people have discovered, I mean, this was known in the late 90s, but people didn't put it all together until about 10 years later, that the following, the bold signal is correlated in different regions of the brain. So here's an example of two uncorrelated regions. I put a seed right there and I extract the bold signal. I put a seed right there and extract the bold signal, and I look at their pattern of spontaneous oscillations over time. These two regions are not correlated. These two regions I'm showing with a circle, they are correlated. Why would they be correlated? The answer to that is, well, for one thing, when that's the terminology here. When two regions exhibit coherent activity like that, we say they're functionally connected. What is the significance of functional connectivity? It's the following. And this, this paper, the first paper about this came out in 1995. But again, the significance of this was not recognized until much later. Here's left motor cortex and here's right motor cortex. If I put a seed in left and a seed in right and extract their bold activity over time, they're highly correlated. One thing we already know is that motor cortex on left and on the right form a functional unit. They're architecturally and structurally tightly connected, and the kind of information they do is they trade information with each other at a high rate. Here's left motor cortex and here's left visual cortex. We already know architecturally and information processing-wise, they don't talk to each other. And their spontaneous bold signals are not correlated. So that's the key idea here. That is that intrinsic functional connectivity, this easily measured bold signal and that pattern of spontaneous bold oscillation is an indicator of network structure of the brain, both architectural and information processing links in the brain. Any questions about that? Yeah, questions already, yeah. Yeah, that's a great question. So one of the first networks to be discovered by means of this pattern of coherent intrinsic functional connectivity was the default mode. But there are many, many other networks than the default mode. So they're not equivalent in the sense that intrinsic functional connectivity is a method of uncovering networks, and one such network is the default mode. And it's good to clear up one other misconception, because I think this is one of the most common misconceptions. Intrinsic functional connectivity doesn't exist only at rest. It's task irrespective connectivity, which means it happens at rest and it also happens during task. It's really important to emphasize because one of the most common questions I get from an audience member is, oh, you're studying connectivity during task. That kind of connectivity is only present during rest. No, no, no. It's present during both rest and task. It's task irrespective connectivity. That's what spontaneous oscillations means. Yeah. Yeah, the idea is that you could put a small seed pretty much anywhere in that region. That's right. So the spatial boundaries of the regions that are exhibiting coherent activity are wider than just a small seed. It would be entire regions of the brain. Yeah, and we'll actually see that in more detail coming up in the slide after this. This phenomenon of this kind of coherence, spontaneous oscillations exhibiting coherence, it was known in 1995. The default mode paper came out in 2001. Much more interest came out in about 2005. And these days, there's literally thousands of papers on this topic, and it's coming out all the time. This has become a kind of a big area of research. I'm going to focus, there's many different ways to come up with a number of how many networks there are. But broadly speaking, many people would settle on about 7 major networks. And this is a recent paper by Yo and colleagues, and they show the 7-network parcelation. Again, what this means is that here's default mode in this red. It's as if the default mode is singing a song. It's singing row, row, row your boat. Every region in the default mode is singing that song. Every region in somatomotor network over here is singing a different song. It's slightly more complicated than that. It's not simply that these networks are coherent within network and not coherent outside, but there's patterns between networks. So that this network default mode may have an anti-correlation with some other network. And the bottom line is that in psychiatry, we believe that a lot of psychiatric disorders manifest by perturbed connections within major networks, as well as alterations in connections between major networks. So this theme of there being networks in the brain that are exhibiting coherent activity within a network and exhibiting interesting relationships with other networks, that's going to keep coming up again and again, which is why I'm making a big deal about it right now. Any questions about that? Okay. Yeah, I mean, it depends on what you mean by how well. There are maps of the brain that you can get from DTI, of white matter tractography, and there's definitely links between functional connections and structural connections as revealed by DTI. There's tracer methods in monkeys, and you can find, and there is ways of showing that functional connectivity lines up with architectural links that we can get from animal studies. So it's not perfect, but there is similarity. Yeah, right. That's subcortical regions, and it's blacked out for the purposes of this parcelation. Basically, we treat gray matter and cortical regions. Excuse me. We treat them in some of these network maps, and we ignore the subcortical regions. And it's not because they lack intrinsic spontaneous oscillations. They do. I don't know the reason why. For most kind of parcelation schemes, omit the subcortical regions. I don't know if anybody else knows the answer to that. Because they certainly exhibit spontaneous oscillations. You can put seeds in them, and you can find their patterns of connectivity with cortical networks. All right. Yeah. Yeah, effective connectivity is, it relates in the sense, in many ways you can think of it as the relationship between correlation and causation. Functional connectivity pertains to relationships that are discerned by correlations between regions, and effective connectivity tries to use patterns of correlations between regions to infer underlying causation using different kinds of methods. So effective connectivity lines up with causality. So again, two regions of the default network are coherent, a region of the default network, and here, violet is ventral attention network. They're not correlated, and in fact, it's likely that these two regions, many people believe, are going to be anti-correlated with each other to a certain extent. So intra-network connectivity and between-network connectivity. Traditionally, connectivity has been studied with seed-based methods. So I put a seed over here. I take out the bold time series. I see what other regions of the brain are correlated with that seed. Perhaps it's this map right here. Similarly for that seed over here. Similarly for this seed over here in yellow. That method gives you windows for particular regions of the brain, but you can't really get a sense of what's going on in the entire brain that way. So Connectomics is the method where, and this is what I'll be talking about today, is basically you put seeds throughout the brain. You develop some scheme of parcellating the entire brain, put seeds everywhere, and then for each seed, you can ask what is its correlation with each other seed. That generates a matrix like this. Since this is correlation and the correlation between this 1 and 2 and 2 and 1 are the same, you can kind of throw out the lower diagonal, throw away the diagonal as well because that's all ones, and then all the useful information about how each seed is correlated with every other seed is contained in the upper diagonal here. We usually use about 1,080 seeds, so this is a very big object. It probably has 600,000 elements in it. If you want to think about it visually, it would be a tangle like this. Every seed's correlation with every other seed would be a huge object like this. This, interchangeably, that thing right here or that thing right there, that's a connectome. And that's kind of going to be the unit that we keep analyzing in all our studies. So let's talk about some of those studies and what kind of things we can do with the connectome. And so every analysis I'm going to be talking about are work undertaken with my colleagues here, all of whom are here, which is why this audience seems kind of filled out. It's many people that I brought. Daniel, Mike, and you are in the Department of Psychiatry. Clay is Associate Professor in EECS. Tak is a graduate student in EECS. And these guys are the brain power behind this operation. So I'm going to be pointing at them to answer questions that I can't answer. And you know, all these analyses were done very collaboratively. Daniel, in particular, could stand up here and give this talk. And I'm sure, actually, he could do a better job than I could. This is all collaborative work. This is the data sets we have. A lot of them come from sharing initiatives. So one of the great things about resting state is it can be done in a standard way across different places in the United States. And so people can pool the different resting state scans they get from patient groups across different places. I'm going to be talking about the ADHD 200 data set, which is misnamed because it has about 800 scans, and the autism data set, which has about 1,200 autism scans. People familiar with our regular studies that we do when we collect our own data, which has 20 to 40 subjects in it, these are big, big data sets. And these are the ones that we're going to be analyzing. So the ADHD 200 data set consists of these sites. There's actually one more site than this, but its phenotypic data was not available. This is actually the data that we analyzed. So we have 462 total scans after getting rid of ones that didn't pass quality checks. 263 ADHD and 462 healthy, 263 ADHD for a total of 725 that we analyzed. So this is the flow of the analysis. This first one is a mass univariate multi-level model. So I'll show how this analysis was done. This is the overall flow, and the overall goal is to get a map like this on which we do statistical inference. But I'll walk through the steps of how this thing works. For starters, we have our subjects at all the different sites. So green is healthy, red is ADHD. Each subject contributes a connectome. Recall, that is, you take their bold time series from the resting state, you calculate pairwise Pearson's R's. There's 1,080 seeds, so you get 600,000 elements. So you get all these connectomes. Each subject contributes one. The next step is we set up this kind of multi-level model. And I'll say more about how this kind of model works in a moment, but it's basically a kind of regression analysis, and we'll get the effective disease out of this model. The next step is based on the significance testing of the effect of disease. We create a thresholded map that represents all the edges at which ADHD was greater than healthy control or significantly different than healthy control. And then we do some additional statistical inference on this map. So let me walk through those steps. This is what the multi-level model looks like. So for each feature, there's 600,000 features, and we're going to do every single feature separately. For each feature, we set up a regression analysis where there's a beta for the effect of disease, a beta for the effect of site, because these data are coming from different sites. Each site has a different scanner. The scanner is calibrated differently, has different signal-to-noise ratios and things. So we need to regress out the effect of scanner. Motion is a huge confound in imaging analyses. So we have the effect of motion. And it's a random intercept model. So we also have a mean zero intercept for site, as well as a regular regression error term. And we do this separately for every single feature. So we do 600,000 separate analyses. This is the map that we get, and we threshold the map at p less than 0.001. And I'll say more about the structure of this map in a moment. One thing to notice is that there's going to be a lot of false positives in this map. Why? We did 600,000 tests. We only thresholded it at 0.001. So I'm going to have to say more about why you should have any faith or credence in the results of this map. Second, red means increased in ADHD. Blue means decreased in ADHD. So we're showing with color the difference between the two groups. The next thing is about the structure of how this map is laid out. The great Japanese computer scientist Takanori Watanabe discovered the use of this map. So the basic idea behind this map is that the edges are now laid out in a way that makes it very clear the network affiliation of the edges. So all the connections within default mode network are now easily seen with your eyes right there. All the connections between default mode network and visual network, for example, are easily seen because they're right there. So we're taking this kind of map that parcelates the brain into different kind of network clubs, and we're imposing that on this map. And that's going to help us organize the information that we're getting when we calculate the differences between the two groups. As a third example, here's ventral attention network and its connection to visual network. Okay. So is this kind of map clear? Most of our data is going to be presented on maps like this. And this is one thing that's been very helpful for us. People are not necessarily using maps like this in the field. And this has been a very helpful thing for us to get familiar with our data and see what's going on in the between group differences. Any questions about this? Okay. So the next step is, if we Bonferroni correct the significance of every single one of these edges, so every dot is an edge, or a connection, or a member of the connectome. If we Bonferroni correct, which means you correct for the fact that you did 600,000 statistical tests, this would be blank. So but the Bonferroni correction is not the right approach here. The thing that jumps out at you when you look at a map like this is that the density of the presence of these dots is not randomly distributed throughout. First of all, there's more dots than you would expect by chance. Why is that? Because by chance alone, because we threshold this map at 0.001, 1/10 of 1% of this thing should be filled, because we expect 1/10 of 1% false positives under the null hypothesis that there's no difference between disease and healthy. But this is more than 1/10 of 1% filled. Not only that, in certain cells, cell just means a place where two networks intersect. So here's the cell that links default network and ventral attention network. Certain cells are filled much more than you would expect by chance. So that's the basis of what we're going to call a network contingency analysis. What that is for each cell, we're going to calculate the number of significant edges observed compared to the number that you would expect by chance alone, which is 1/10 of 1%. And the actual test that we use is we use a binomial distribution with parameter p.001, and n is the number of edges in the cell. The result of that, and now shading is being used to show where there are cells with statistically significant more edges than you would expect by chance. And much of this map is filled at this point. So what that means is that between disease and healthy, certain regions of this map, in particular the ones that are shaded, are highly statistically significantly different. That is, those cells have far more edges than you would expect by chance. So even though Bonfori correction applied to the individual dot, it is not statistically significant. When it's applied at the level of the cell, there clearly are very big differences between these groups. You can actually see the p-values here are ranging between 10 to the negative third and 10 to the negative 15th. So some of these are absurdly high p-values. So what is the interpretation of this? Here's the default mode network. There's hypoconnectivity in ADHD within the default mode network. Here is the ventral attention network. There's hyperconnectivity within the ventral attention network. I'm sorry, I said that incorrectly. There is hyperconnectivity between the default network and the ventral attention network. So those two networks are more correlated, or another way to put it, they're less segregated than in healthy controls. That's an important finding because that's one of the most widely discussed and replicated findings in the field, that in ADHD there is enhanced connectivity between default mode and dorsal and ventral attention networks. So this map is kind of showing you things that the field already believes, but showing you from a different perspective. Any questions about this map? Yeah. Yeah. So a qualitative answer to that is the structure of resting state networks is thought to be very reliable and robust across individuals and within the same individual across time. So yeah, I can't give too much more quantitative information beyond that, other than it is thought to be reliable and robust. Yeah. Question? Yeah, I should have said what that means. Yellow means, there's one thing I didn't say, and that is, in these cells, when blue predominates, we shade the color of the entire cell. We do a sign test, and we shade the entire color of the cell based on what color is predominating in terms of the edges within the cell. And yellow means it's mixed. So that there is, by the test of, are there more edges in this cell than you would expect by chance? Yes. Is the directionality of the difference predominant in terms of enhanced or decreased connectivity? No, it's a mixed bag. That very same cell is showing a mixed picture of hyper and hypoconnectivity. That's what the yellow means. To put a map like this in context, I think that we were very, very surprised to see this map. And by the way, we did this analysis maybe 2 weeks ago. So it's still kind of sinking in the meaning of this map. We usually don't see highly significant results across the entire brain in our fMRI analyses. Usually we're trying to coax signal out of the data. We certainly don't see effects at 10 to the negative 15th. I've certainly never seen that. Now, one thing is, we don't work with data sets that are 1,000 subjects. We work with data sets that are 40 or 50 subjects. It may very well be the huge effect sizes we're seeing is because we have so many subjects. But it's also likely that this kind of connectomic map is identifying discriminative information in the data that wasn't present otherwise. And one way we can kind of say that is when we do connectomic maps on normal-sized data sets, we also get pretty inflated p-values. So I think that we are getting more discriminative information out of the data with this kind of analysis. Daniel, do you recall? It's all over the place, probably. Okay. Yeah, let me, a couple of places I'm going to put questions out here. I mean, one of the reasons why this talk was so attractive for us to do here is because we thought there might be people in the audience that would answer, questions that we have about the data. And one of them is, and so, Hamid is here. Hamid is Al Hero's graduate student. We talked about hub structure, hubs and other motifs that might be in the data. So we're analyzing this with kind of, univariate tests that are done separately of every single edge. But clearly this kind of data is susceptible for identifying higher order features, graph theoretic kinds of approaches. It just jumps out at you with your eyes that there are regions. So this node right here is changing its connectivity with every single other node inside the default network. Here's another kind of hub area. There's a node somewhere over here that, at least in the frontoparietal and default network, is changing its connectivity with a lot of other regions. But the hub structure, so a hub is a region that's changing its, or has affiliations with a bunch of other regions. The hub regions are moderated by network here. So it's not as if the hubs, I mean, maybe some of these hubs are across the entire brain, but other hubs seem to be network specific. So somebody needs to devise a method to look at this data and assign a p-value to the hubs and correct for multiple comparisons. And so then we could, write up and say, in ADHD, there was a significant difference in the hub status of that particular node right here, and that's a highly statistically significant result. We need to be able to say that, and somebody needs to tell us how to do that. And I think, there's clustering methods that are available. Our a priori parcelation using the Yo network does seem to capture structure in the data. That is, there's clearly a lot going on inside default network. The default network is an a priori categorization that we impose on the data from the Yo network. But there may be better parcelation schemes, or there may be data-driven parcelation schemes. So I would say that those people in the audience that have a background in some of these methods, you should ask yourself, what would I do if I had a connectome like this? What would I try to analyze it with? And if you have any ideas, you should let us know, because it seems like this is a very rich data to study. So that was the multi-level model, the network contingency analysis multi-level model. I now want to show the data on autism. For that, we have both a multi-level model as well as some classification results. So the autism data is actually even bigger and more complicated. I don't know, do you remember how many subjects we included? This is how many subjects are available on the site, and I forget how many we actually included. It was like 9, about 1,000? Okay. And these are all the sites. And this is all publicly available data, by the way. So it's the same kind of thing over here. There's all the subjects across the different sites. It's not just 2 sites. I'm just showing 2 for ease. Every subject donates a connectome to the analysis. We do a multi-level model with the effect of disease as well as a random intercept term. We threshold it at 0.001, and then we do the network contingency analysis to do statistical inference, which gives us the shading. Here's what we see in autism. Once again, it looks like a quilt. It's kind of an abundance of differences, and there's just no other way to put that. There is tons and tons and tons and tons of differences between autistic brains, and once again, the p-values are through the roof, up to the 10 to the negative 15. There is a lot of diffuse hyperconnectivity between these regions, default and frontoparietal, and multiple other regions. Intra-network connectivity is almost uniformly hypoconnected in autism. And it's just hard to summarize the quantity of these findings. There's a lot of them. For those of you that study autism, you'll recognize that pretty much aberrations in every single network have been identified in autism, separately with seed-based analyses. And that's kind of what we're saying here. if you had done seed-based analyses, no matter where you put your seed, you're going to find network abnormalities, except we're showing this connectomically now. For comparison, here's ADHD and autism side by side and back to back. ADHD has a lot more hub structure in the default network. That's one big difference. And in fact, the hypoconnectivity is far more diffuse and heavy in ADHD than autism. One big difference is in somatomotor network, which is clearly hypoconnected and has hub structure in autism, but is hyperconnected and lacks hub structure in ADHD. So one thing is clear. There's different kinds of network abnormalities in autism and ADHD. It's not the very same thing going on in the two disorders. We've also done schizophrenia, by the way, and it's got its own pattern that looks very, very different than these two. We've never done case against case. We've never run that. Well, I just said that we had done most of these analyses in the last few weeks. This is all fresh off the press, so that's a great idea. It's to be done. Yeah. 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 I think that's right, and I think that we really need to also show that there's reliability in the signature at the level of a single individual, such that you could predict their disease status based on a connectomic map. And that's what I'll be showing next, that we can do that to a limited extent. And I think that would kind of address your concerns about heterogeneity. Not much. So what we we do know from other studies that have looked at healthy individuals as well as patient populations separately and in small groups that there is good retest reliability in resting state connectomes across time. So, you know, how good is good? Good enough for science, I guess. Yeah. 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 Yeah, that's a really great point, and I think that these results I think are believable, but they'll be really believable if we permuted the labels 1,000 times of autism and healthy, and we showed that these p-values are pretty extreme on the permutation distribution. That would be pretty convincing. That's a computationally intensive analysis, and when CCMB supports us with another grant, we will be able to. No, I'm just kidding. That was yeah. Go ahead. Yeah. Yeah. At one time, TAC had done a MDS multidimensional scaling on the data. That's another way that you can recover interesting groupings in the data. I'm not an expert in these methods, but there's a lot of these kinds of data-driven clustering and grouping methods. I'd love to try to do them on these big data sets. Nobody else has done them yet, these analyses. So it's a really good idea. Yeah, I just realized there's only 10 minutes left. No, that's fine. So I'm not going to get to the test data. I think I'll just walk through the classification result, and we can stop there. Here's the classification analysis. This is the structure of it. So again, every subject contributes a connectome, and then then we're doing classification, and we're going to visualize some aspect of the information the classifier used. Here's the key step: the feature pruning. Now, with 600,000 edges, if we stuff them all down the mouth of the classifier, its performance would be poor. We need to prune that down to get to be the most useful features. That's where we're using the graphical lasso. So this is a novel, kind of interesting way of pruning the features. So I'll walk through how these steps work. By the way, we're doing the feature pruning and classification embedded within a leave one out. So it's not this kind of biased analysis where you did that on the entire data set. This is the Glasso, and this is where Tak and Clay and Daniel will know the details of it. But very roughly what we did is we did separate analyses on the healthy. We concatenated their time courses, and we got a mega time course. Then we did Glasso on it. What is Glasso? It's the objective of the Glasso is to find a sparse inverse covariance matrix. It's a matrix of partial correlations. Very roughly, you can think of what Glasso is doing is it's finding the non-redundant pieces of information in the full edge set. So the connectome is full of redundancy. There's lots of intercorrelations between edges that are doing the same thing. If you control for all the other edges, maybe one connection would disappear. That is, its partial correlation would go to 0. The Glasso is going to find the non-redundant edges of the connectome. So using Glasso, we get a sparse representation for the healthy, a sparse representation for the disease. Then we take the XOR, which means we're looking wherever the Glasso had said there's a non-redundant edge over here that's present in healthy, it's not present in autism, or vice versa. That gives us a kind of a screening matrix that we then impose on the connectome. And those are the edges that enter the classifier. So here are the results. I'm only showing the big 4 data sets, the data sets that roughly had 150 or more subjects combined between autism and healthy. So at NYU, classification was 92%, and here's the breakdown between healthy and ADHD. Over UCLA, 86%, U of M, hometown disadvantage, 72%, and Utah, it was 82%. Those are good numbers. That's good. It's reliable across each individual site. And the Glasso maps, to the eye, they don't look that similar, but I think you might make out certain kinds of things, especially in UCLA, NYU, and University of Michigan, that are similar. The alternative to the Glasso, the more standard method is a univariate t-test for feature pruning, and we have a boost in classification over the t-test at all 4 sites. So Glasso is performing better than the univariate t-test. So I think this method is contributing something new and is showing features of the data to the classifier. So you can probably build a better classifier with Glasso. I think that's the conclusion. But here is the problem. So this is another great problem for anybody that gets interested in this kind of thing. This is a kind of cross-site generalization. This is a map that Tak had produced. The diagonal is what I showed earlier. That is, you test and train on the same site. But what if you train on one site, get the Glasso feature pruning matrix, then put that matrix on another site's connectome and train an SVM classifier? Well, the generalization is not that good. Why is that? We call this the multiple scanner problem, and it's an underappreciated problem in the field. One of the reasons we're able to build a good within-site classifier, but a poor cross-site classifier, is because the kinds of noise that's contributed by scanner, problems in co-registration, problem in signal and noise properties, and things like that, we're not adequately correcting for that in our usual methods. So that's a great unanswered problem in the field. If sharing initiatives are great, they're outstanding. They bring together a lot of data. But until you solve the multiple scanner problem, you're not going to get a really great classifier that works across sites. I mean, Michigan's, well, we don't have a good answer for that, A. B, it is a different scanner. Well, the Michigan is lower within site as well. So, yeah. So oh yeah, Michigan only has one scanner, yeah. It's the GE. I wonder if our signal-to-noise ratio is not as good as some of the other places. I also wonder, we clearly had more motion in our subjects here in Michigan than elsewhere. And we've seen that in a lot of data sets. Our motion tends to be higher. I wonder if we need to be doing more things like giving people a bite block or something like that to keep their heads still. I don't have a good answer for that. The last thing to show was cognitive tasks, but we don't have time for that, so I'm just going to... All right. Really? I'm going to go over. What do you think? Should I wrap up or do the last segment? Okay, I'll blaze right through this. Cognitive tasks. So everything I've talked about so far is biomarkers of disease. There's no doubt that that stuff is important. But another huge thing that we do in fMRI is we try to figure out the mechanisms of cognitive processes that are relevant to disease. And it turns out Connectomics can be very, very helpful and revealing in this area. And I'll show briefly how to do that. In resting state, it's easy to get the entire time course for the entire experiment. In task, what we do is essentially what we do is we chop. So here's a task. I give people pictures of faces, and I give them pictures of houses. And I'm interested in the mechanisms that, how brain regions are communicating with each other when they're seeing faces versus the control condition of seeing houses. For those that think this is a silly example, faces, houses is the usual toy example when you're explaining an fMRI task. It's just a silly example that everybody uses. So I take the time series that's face-specific, concatenate that, and generate a connectome, just like I did in the resting state. I take the time series that's house-specific, concatenate that, and generate a connectome. So, you know, I've seeded the brain, generated a connectome for faces, one for houses, and then I take the subtraction. Every single subject is contributing a face connectome and a house connectome and a subtraction connectome, and I analyze it just the way I did before. And I come up with one of these shaded talk graphs. By the way, we call these graphs talk graphs after the great Japanese computer scientist Takanori Watanabe. All right. Emotion regulation. So in psychiatry, we care a lot about emotion regulation. My own pet hypothesis is that there's a lot of people walking around with emotional abnormalities. They feel too much fear or too much sadness. The ones that have disease are the ones that don't have the right kind of ability to regulate the emotion process. So two hits need to happen. You need to have aberrant emotion responses, and then you need poor regulation of it. So regulation refers to cognitive voluntary modulation of your own emotion responses. We can test that in the scanner. We give people harsh pictures like this picture over here. We ask them, just react to that picture the way you normally would. Just maintain your reaction. In other trials, we give them the same kind of harsh pictures, and we ask them to reappraise, modulate, reduce your internal emotional reaction to that picture. And so the subtraction between reappraise versus maintain is those cognitive processes that are involved in emotion regulation. In an activation analysis, for those that have seen these kinds of results, this has been done 30, 40, 50 times, many published studies. This is the classic map you get. Dorsomedial prefrontal cortex is activated during regulation. You also get dorsolateral prefrontal cortex here and here, and then kind of inferior parietal regions and dorsal attention network here and here. Everybody has seen this picture. Very, very common result. Here's the connectomic result. So just focus on the top half. What you're seeing is that during emotion regulation, the default network becomes much more connected to the visual network. At the same time, the dorsal attention network becomes more segregated or anti-correlated with the visual network. So connectomically, we're seeing the mechanisms of connectivity during emotion regulation, and that's a very hard thing to see otherwise without using the connectomic methods. Why are there two maps? Because task-based connectivity is not fully reversible. What you get is two maps that are very similar to each other, but they're not identical. So we have to show them both and kind of establish that they are similar. And sure enough, they are. Default network and visual, and dorsal attention and visual are the key findings in the top map as well as the bottom map. So I want to put it out there that connectomics is not simply for disease identification. Connectomics is a very general way of analyzing neuroimaging data. And these p-values are, really huge, like we saw before. And this is only 40 subjects. So connectomics is pulling out a lot of very useful signal in the data and showing you perspectives on the data that you wouldn't see otherwise. In conclusion, in psychiatry, we want to develop biomarkers. Traditionally, that's been done with activation analyses. Connectomic imaging is a new approach. We've done studies in ADHD and autism and showed that connectomics is showing very interesting network abnormalities in these disorders. It can also show mechanisms of cognitive processes during task. Once again, I want to thank my colleagues and collaborators, and thank you all for your attention.
ADHD and then autism, were any of them on medications at the time? And does that play a role, do you think, in how this connectivity might be modified by pharmacological agents? I don't think it would be advisable IRB-wise to treat people prior to this, but if they're already on medications, is there a way to correlate that and look to see if there's modifications based on pharmacology?
So with the online data sets, we don't get as rich phenotypic characterization as we typically get when we have our own data sets. So the problem is the online data sets does not include history of previous medication. In ADHD, it's usually not that big of a problem. The reason is that stimulant medication probably does not leave long-term signatures on the brain. The one that I really worry about is schizophrenia. It's almost impossible to get clean data from schizophrenia with people that are not medicated. And the online data set on schizophrenia does not contain medication status. So this is a real problem. These online data sets are great in terms of the number of subjects you get, but the phenotypics on them is kind of skimpy.
More questions?
Question.
I was struck in the cognitive test that the blocks for red and blue were a much smaller part of the total picture than for the diseases.
Yeah.
Is that a big conclusion?
So there's many things that could be going on. One is, in a way, these are within subject analyses where each subject is their own control. I bet you that we probably have better ability to suppress signal and noise in a within subject design like this, A. B, there's far fewer subjects here, so that could contribute to why certain weak signals are not found. And C, it is plausible that cognitive processes like emotion regulation affect discrete regions of the brain, whereas diseases like autism, which has been conceptualized as a diffuse hypoconnectivity disorder or altered connectivity disorder, would have massive signatures all over the brain. So that's at least 3 reasons why these two things could differ. And also I should say that task-based connectivity and resting state connectivity, we don't have a good feel of how those two differ because there's very few task-based connectomic studies. Ours could be among the first or the earliest. Task-based connectomic studies, I should say. There's plenty of task-based connectivity. So these are all reasons why those two could differ.
Do you think we'll ever get to the day when many mental disorders will be diagnosed by imaging functional connectivity?
Yeah, that's hard to say. I think we'll get there. And one thing I will point out is that I used to be the director of the ADHD clinic here in Michigan. It's a tiny little clinic that operated once every 2 weeks. We kind of closed it down because there wasn't enough people to operate it. Our standard way of diagnosing ADHD is with neuropsychological tests. Are there any neuropsychologists here? Okay, then I'm gonna be insulting about neuropsychological tests. It's clear that they have no predictive value whatsoever. So you send people to neuropsychology, they do 4 hours of testing, and the literature clearly shows, and this is very expensive testing because it's half day of testing. These tests are well designed. They're thought of by very serious people. But if you actually look at how much discriminative information they contribute, it's none. We're already better than that with this stuff. I mean, so these 82% detection rate, yeah, it's a little bit inflated because we've taken a very refined sample, people with ADHD, comparing them to people without, and we've changed the base rates from what you might see at a clinic and things like that. But these tests are probably better than the kind of things we're doing right now. So, yeah, so compared to the crappy things we're doing right now, yeah, I can see a day when we use things that are a little bit less crappy. Sure.
Okay, this is really a very interesting talk and generates a lot of discussion and questions. So let's thank Chandra again for the fantastic talk.
Thank you. You know that thing about neuropsychology? Let's chop that off the video.